We will be looking at cell division and cell cycle, that is chapter 10 from grade 11. Firstly, let's talk about cell cycle. In a cell cycle, there are 4 stages. Stage number 1 being G1 phase, then G2 phase, S phase, as well as M phase. So, this is how you might think the cycle to be, but then the order is shuffled. It is G1 first, then S, then G2, then comes M phase. So, what are the various things that happen during these stages? During G1 phase, the cell grows. So, this is also called as the growth phase. Because during G1 phase, the cell grows in its size, in its width, etc. What about S phase? During S phase, the full form of S phase is synthesis phase. So during S phase, definitely something gets synthesized. What is synthesized? DNA. DNA is synthesized during the S phase. In G2 phase, that is gap 2 phase. This is gap 1 phase, synthesis phase, and gap 2 phase. During gap 2 phase, proteins are synthesized. So here growth happens, here synthesis of DNA happens, here proteins are synthesized, as well as cytoplasm, that is the fluid matrix of the cell, begins to grow. It continues growing until the cell enters the mitotic phase. So what is mitotic phase or M phase? So during the mitotic phase is what cell division happens. So cell cycle is the combination of G1 phase, S phase, G2 phase and mitotic phase. The mitotic phase is split into 5, with number 1 being prophase, number 2 metaphase, number 3 anaphase, then comes telophase, and then finally cytokinesis. So, what are prophase, metaphase, anaphase, telophase, and cytokinesis? These are the 5 divisions of the mitotic phase. So, these happen during the mitotic phase. This is when the cell starts splitting. So, after the growth of the cell is done, and then synthesis, the DNA synthesis, as well as protein synthesis, as well as cytoplasmic growth is finished, the mitotic phase begins. So during that, first is prophase. What is prophase? Suppose this is a cell with its cell membrane. There is DNA, there are chromosomes like this. During prophase, the main changes that happen is all the cell organelles as well as the cell components, that is cell organelles, starts to condense, or it just disintegrates. It's no more seen inside the cell. And all the chromosomes or all the chromatic threads condense to form chromosomes. So this is how chromosome looks like. So it combines to form a chromosome. So this is the main changes that happen during prophase. Next is metaphase. During metaphase and in prophase, there is a component called as a centriole. 2 centrioles, that is a centriole duplicates and it comes in one of the poles and the other one at the opposite. So it comes at both the poles. Its role is seen in the metaphase. During metaphase, the cell with the cell membrane with the centrioles start producing thin fibers. These thin fibers are called spindle fibers. So in this there might be chromosomes. Let's take 3 for this example. 3 chromosomes. So we know in a chromosome there is a centromere, as well as on the sides there are kinetochores, right? So the spindle fibers comes and attaches to the kinetochores of the chromosome, and these get aligned in the middle. Let's take it as the equator of the cell. So they come and align at the equator of the cell, or we can tell it as the equatorial plate. It is also called as the equatorial plate. So that is metaphase. So we did prophase, we did metaphase. Next is anaphase. What happens during anaphase? During anaphase, the centromeres, that is the middle part, split. So if this chromosome splits into two, we'll get two different centrids. We call this as centrids. Chromatids. We call this as chromatids. Okay, so in anaphase, the chromosomes split into chromatids. This is what happens in anaphase. The main thing that happens in anaphase, the chromatids split and it moves to opposite poles. So the spindle fibers keeps on retracting until all the three, or how many chromosomes or centroids are there, it moves to the opposite side. So that's what happens in the anaphase. Next, telophase. What happens during telophase? Telophase is the time when the chromosomes cluster at opposite spindle poles and their identity is lost, and the nuclear envelope starts developing. So it's just the opposite of prophase. The cell components becomes more, seen. The nucleolus, Golgi complex, and then endoplasmic reticulum reform. So during telophase, let's draw a diagram. So this cell starts kind of splitting like this. We can see the centriole still there. This is what you'll see during a telophase. So the centrioles start disappearing and then the organelles come back, and then finally the cell splits. So this is what happens during cell division and cell cycle. So once again, cell cycle comprises of gap 1 phase, or where the growth of the cell happens, then the synthesis phase where DNA is synthesized, gap 2 phase where the proteins are synthesized and cytoplasm grows, then mitotic phase where prophase, metaphase, anaphase, telophase and cytokinesis happen. Number 1 is prophase, when the complexes or the organelles disintegrate and centrioles split and move to the opposite poles. And then the condensed chromosomes form in the middle, also called as the equatorial plate, with the help of spindle fibers which are attached to the kinetochores. Then anaphase is when the chromosome's centromere splits and then the centrids move to the opposite poles. Then telophase is when all the organelles reform, nucleolus, ER as well as Golgi complex reforms, and the cell splits. The splitting of a cell is called cytokinesis. Karyokinesis is the splitting of nucleus and cytokinesis is the splitting of cytoplasm. I hope this video helped you a lot. If it did, please like and subscribe to the channel. Thank you for watching.